Oh boy, so now we move into a really, really big chapter, chapter 33. I say really big chapter in terms of the stuff we're going to talk about and how much we're going to talk about. There's just a bunch of invertebrates we need to go through, and we need to discuss the differences between them and put this all in the context of what we put everything in context before the semester, and that evolutionary significance. I found this really neat picture of the insects there in the lower left. I think to me, it just demonstrates the diversity we see just of the insect group, let alone the rest of the invertebrates, just the color and the shapes and the forms. I read somewhere one time that folks have the heebie-jeebies about insects many times because of the fact that they have these jointed legs. See, like in that picture, the close-up of the wasp there on the right, that's really what it is. It's the jointed legs that they move around. I wonder if that's because it kind of reminds us of our ability to move around. And insects and arthropods, which we'll get to at the very end of this chapter, became very successful because of this ability to move around and diversify. So again, we're going to kind of take that concept of the fittest and diversification and expand it into like a phylum level when we talk about the arthropods. There's just so many of them, but we'll get to those in a little bit. Let me continue on talking about just the invertebrates in general. Of course, I'm sure you're all aware the invertebrates are those organisms that do not have a backbone. There are vertebrates, and then there are invertebrates, and then, of course, like everything else, this semester there's a few critters that fall somewhere in between. So we'll get to those when we do. 95% of all the animals out there, 95% of all the critters that we consider animals are invertebrates. So it's a really large group with a lot of diversity. If you think about it, it's significantly more diverse than the vertebrate group. The vertebrate group is the chordates right there. There are some groups that are kind of in between again, but that's the vertebrates. That's you and me and dogs and everything else, right? So look at all the other groups. Those are the invertebrates. And we start up at the top again. I showed you this in class. I should correct this slide. That should not go around. Diploblastic should not go around periphery. Diploblastic refers to two tissue layers, two embryonic or germ tissue layers. That's what ectoderm, mesoderm, and endoderm were that we're talking about. During embryonic development, those are the first layers that separate out in tissues, and then those become things, right? They become the muscles or the skin or parts of the skeleton. Organisms that are diploblastic only have the outside, the ectoderm, and the inside, and the rest of us are triploblastic because we have the middle part, the mesoderm, right? Periphery can't be considered diploblastic because of the fact that it does not have tissues, right? Periphery is a multicellular aggregate, which is kind of bordering on tissues, but not yet defined. Remember we said tissues in animals have a very specialized definition. They refer to a collection of cells that perform a very specialized function, a functional unit, right? Like nervous tissue again. Don't find those in periphery, so that's not correct. The ctenophorans and the cnidarians, which is the first two groups that we'll talk about, do have tissues, and they're considered diploblastic. So indeed, they fall into the group that I pointed out to you yesterday, known as the Eumetazoa. Try to remember what these words mean. Literally, if you break them down, it'll help you remember them for this class and in the future. Meta is more than one, right? Meta, mega. Zoa refers to animal cells. So a metazoan has many cells and is an animal. A eumetazoan, EU means true. Oh gosh, what's the one in Bio 1? Holy cow, I just drew a blank. In Bio 1, there's a U. Eukaryotic, there you go. Eukaryotic means a nucleus exists. There is a nucleus. So eumetazoa means, roughly translated, is many animal cells truly. In other words, tissues. All right, eumetazoans are tissued organisms. So let's go with that. First, let's go with the eumetazoans about 680 million years ago. So this is before the Cambrian explosion. These are the critters that were around. Remember, the Cambrian explosion is when a lot of the rest of these come around. So let's move now and do the cnidarians and do the ctenophorans. I think we do the cnidarians first, if I remember the order of my slides. The cnidarians. Oh, I'm sorry. Poor sponges. I totally skipped them. Let's go to the peripherans back for a second. Out of the eumetazoans into the multicellular, but not tissues, no organs, no symmetry. May look like it, but no symmetry. Most sponges are going to be found in the ocean. There are a few freshwater species. The ones I'm aware of are very small, though. The freshwater species are like not microscopic, but not very big at all. A variable growth form, which goes with the fact that they don't have symmetry, right? Even though that vase, that purple vase sponge there, kind of looks like it's symmetrical, it's only because of the way it grows. There's many sponges who grow blobular, if you will. As a larvae, though, and this kind of goes back to this idea of dispersal, it's amazing to me as we go through and talk about the animals, the parallels that I hope that you guys, or that I draw, and I hope you guys can draw. It's amazing to me the parallels between how animals evolved as the earth changed and how plants evolved, and how anything evolved, and this whole idea of variability and dispersal, and you know, so you don't compete with the parents for resources. So among the sponges, which are sessile, right, not immobile, they settle down and they grow and they don't move. Sponges don't walk around anywhere. They don't swim around. Except when they're a larval form. In the upper right, see that little picture? That's a larvae of a sponge. It's microscopic. It's just looks like a drop, kind of, right? Is planktonic. Now remember the word planktonic, I think I mentioned to you once or twice before. I'm sure planktonic in the ocean refers to things that float. It's a term that's easy to confuse because there are things called plankton, phytoplankton, maybe you remember some marine science class, zooplankton, and these things are generally microscopic, but there are even fish that are plankton at certain times of their lives, when they're larval, for example. So a sponge can be considered a plankton during the time when it's a floater. Plankton is to float. The other word in marine science is nekton, or nectonic. Nectonic is to swim. So fish are nectonic, and these things are planktonic. They float, right? Make sense? And then these larval forms that are planktonic drift with the currents and then eventually settle down into an adult. So that's how dispersal happens. Poriferans are relatively abundant in the deep ocean. The one you see in the lower left, that really cool picture of that sponge, it's actually a sponge. It looks like a spaceship kind of thing. That picture was taken at over 2,500 meters in depth. So that's way down there. Not something we see parallels in deep ocean organisms is not, I tell my marine science students, not fighting back at the pressure. In other words, don't have rigid structures, be blobular. A lot of deep water fish, really deep water, have very flabby muscles, flabby skin. So that's kind of the way they deal with the pressure. So poriferans being, flabby, well, not rigid, would do well in deep water. That's why you often see them there. Okay, let's get into a sponge and cut it open. So in the picture, we've taken just your typical sponge, and they vary, but all sponges are going to have this collection of cells. They're going to have these pores. They're going to be porous, right? That's nothing outside of the realm of what you haven't seen. And then there's usually going to be one or several osculum. I don't know what the plural of osculum is. Maybe it's oscula. But some sponges will have lots of these things. And the water moves into the pores and out through the oscula. And water moving in through the pores, out through the oscula, the water gets dumped in that place that's in the middle there, right? In that space inside of the middle of the sponge. This thing we call the spongocoel. See how the benefit is of learning those words? Seal is space, right? Blastocoel, space. So the space in that sponge, that cavity in the middle of this particular one, is the spongocoel. Now, water moves in. Why? Because there's little tiny cells around the spongocoel that are beating their flagella. And you know what happens when you beat your flagella? Either you use it to move or you move things around you. We've seen that all the way back in Stentor, right? Stentor was a filter feeder. These organisms are filter feeder. They take these cells with flagella, these collared cells, these choanocytes, and if you're thinking, why does it look familiar? Gosh, I hope it does, because those were the choanoflagellates from yesterday. Remember, choanocytes and choanoflagellates are just kind of down the block from one another. Choanoflagellates are protozoans. Choanocytes are cells of an animal. Boy, we're getting right there in the middle, right? The gray area. All right, so all these choanocytes are beating their flagellum. That's moving the water through, and they're using their collar to catch food, right? That's what they do. They're filter feeders. And the osculum then at the top is where the water exits out of. If they capture food with the collar, what they do is they engage in, this is why you can't forget your bio one again, they phagocytize the food. They engulf it, right? Now, here's what's kind of cool about sponges. They're a little bit like the Volvox that we saw back in when we were looking at green algae. Is it one is it a colony of cells, individuals? You know, where does one, you know, where does it end being an individual and then you're a group? You know, that kind of thing. That discussion I've had a couple times with you. Sponges are ones that kind of straddle the fence on that again, right? The cells work together, right? They're not tissues though, but they work together. For example, after the choanocytes have phagocytized the food, which they then use some of the food obviously for themselves to keep their cellular machinery operating, but the rest of it they pass to amoebocytes. Now amoebocytes then take a little bit of that cellular energy as well and use it for themselves, but what they don't use, the amoebocytes move throughout the sponge and they pass on that food. Isn't that cool? So amoebocytes actually act like, I'm looking for the analogy, our blood system in a way, maybe, right? Moving nutrients around. And again, these are not tissues, but cells that are certainly cooperating, aren't they? Isn't that neat? And this is an animal, and very similar to what we would see in like a colony of those protozoans. It's so close, relatively speaking. Okay, on the outside, the anatomy, there's an epithelium or an epidermis. The middle layer is the mesohyle. I love that. Always sounds like a Jewish word, mesohyle. The mesohyle is the middle part of the sponge. It's this gelatinous or a matrix. A matrix is a collection of things that are put together. Like cement is a matrix. It's a mixture of sand and concrete. Right, or is it the other way around? Anyway, so a cake is a matrix. You put flour and water, and you know, why do I keep picking things that I don't know what's inside of them? Whatever is inside of a cake when you put it together, you get the idea. In this mesohyle, there's this gelatinous matrix, right? And inside of that, there are these spongin proteins, and they're spongy. Right? Proteins, remember, are kind of stretchy anyway. But even though they live in the water, they still have need to kind of be held together. This need we talked about when we were talking about animal cells and their cytoskeletons and plant cells and their cell walls. The way they do that, if you look down below the little pictures in the black and white, those are electron microscope pictures of things called that are embedded in the mesohyle called spicules. Spicules are made up of calcium carbonate. They can be calcareous or siliceous in their makeup. Siliceous is glass, basically. Calcareous is shells, calcium carbonate, the coral. We've talked about it before. But whatever they're made of, these spicules are rigid or hard. So you see the sponge gets its shape being held together by this gelatinous matrix of spongin with all these spicules embedded into it. What's really cool about it, too, is the spicules vary the way they look. You can even see in the picture, see the ones on the right are just basically straight. And folks who study sponges, many times the spicules, the shape of them indicates what species or what group of sponges that belongs to. So in other words, the shape of your spicules could be genetically determined. It depends on who you're related to. It's really kind of cool. I talked yesterday about the fact that we can take them with a cheese grater and cause them to asexually reproduce by fragmentation. Now think about an organism that lives the way it does too. Oftentimes sponges live in the tidal zone where the waves are breaking, and getting broken up is something that happens to them a lot, right? Or animals nibble on them and that sort of stuff. So being able to fragment, being able to asexually reproduce this way is probably something that would have been beneficial evolutionarily speaking. But like all animals, they also, well, not all animals, but like most all animals, they also engage in sexual reproduction. right? Most sponges are hermaphroditic. Now we don't, in the sense that they're able to produce both sperm and eggs. Typically, though, like we were talking about in the mosses, they try to avoid fertilizing themselves. So specifically what happens is there are colonocytes inside the spongocyst that during the reproductive time actually transform or become sperm, and they then are moved out through the osculum with the water flow where they drift in the water. Now, most of them don't make it, right? I hope that's the first thing you thought of when I said that. Oh, I'll bet most of those sperm don't make it, and they don't. But you're going to make a lot of them, aren't you? Somewhere down the road, there's another sponge that's going to take in water for food and some of those sperm. Now, when those sperm are passed to the colonocyte is going to pass the sperm to the egg, which is in the mesohyle. Fertilization is going to occur, and check this out. This is cool. The young, the junior sponge, actually begins its development inside the mother in some species. Not in all of them. Some of them, they shed it out into the water. But even in the not lowly. I remember my zoo professor used to say they do so well with so little. So even the ancestral sponge has got this idea of protecting young. Not something that just animals did. Remember, angiosperms did that too. But you notice how there's this shift as we look at organisms as diversity increased in many lines, not all, right? But in some lines where we went with the idea of let's have fewer young, but kind of give them a better chance to survive. And we all fit on the scale somewhere up and down the road there. All right, so sponges are cool, but we got to move on to the true tissue folks now. So let's go with the ctenophorans first. The C is silent in both of those words, ctenophora and cnidaria. Ctenophorans and cnidarians. So let's do ctenophorans first. Ctenophorans, you may have seen these. Sometimes I have actually seen these at the beach out in Sarasota on some summers when I've been out there before. But these are what we call comb jellies, sea walnuts, sea gooseberries. They look like jellyfish, and they actually used to be considered that. Although, as you see in the slide there, how they're related, the phylogeny right now is uncertain. They are in a different group, though, different phylum. They're not in the cnidarian phylum where the jellyfish are. One of the big differences between them and jellyfish is even though they have these tentacles, on these tentacles there are these rows of comb-like fused cilia, and along their bodies as well. They beat in this coordinated fashion, and that's how they move. Some of them are bioluminescent with these bacteria that live within the tissue of the ctenophoran, and that's why I say sometimes at night you might have seen them. You could see the bioluminescence sometimes in the comb jellies. Now they have tentacles, like I said, and they capture their prey. But whereas cnidarians, we're all familiar with this in Florida, the jellyfish and those, they sting their prey. These organisms, in a really bizarre kind of convergent evolution, they don't have these stingy tentacles. They have tentacles that are covered with things called coloblasts, and coloblasts secrete this discharge, so they stick to their prey. So rather than stinging them, what the cnidarians do, these guys gather their prey up by these sticky arms. Really beautiful critters too. Look online and YouTube. There's tons of videos of comb jellies and them bioluminescing. There's one in the lower right bioluminescing. So when these comb-like plates of cilia fuse, they fuse in a wave, like you've ever been to a football game, right? And everybody does the wave. These waves are moving this organism, but these comb-like cilia have these bioluminescent bacteria in them. And I don't know if I've told you before, or I can't remember, but bioluminescent bacteria bioluminesce when they're agitated. And beating them around would cause them to agitate. So when the cilia or these fused cilia beat, the bacteria get agitated and they glow blues and reds and all different kinds of colors. It's beautiful. Okay, so that's the comb jellies. Let's move on now to the true jellies, or the cnidarians, right? So the cnidarians are mostly a marine group. There's a few freshwater. Ironically, you're probably more familiar with the cnidarian that's a freshwater than any other one. That, like in the picture, Hydra, the one you might have seen under a microscope in high school or maybe in Bio 1. Hydra is a cnidarian, and it's actually a freshwater cnidarian. So Even though there's a few species, it's one of the ones we know best. Cnidarians sting things, so they're meat eaters. They're carnivorous. They have radiosymmetry, 2 body layers again. The endodermis is going to become, specifically in them, the part in the yellow in the picture down below called the gastrodermis, which, if you look at that word, you can see what that's going to do is that's part of the digestive system, right? That's where the digestion is going to occur in the gastrodermis, or the gastrovascular cavity surrounded by the gastrodermis. And the outside's the epidermis. No organs in these folks yet. We got these beginnings of tissues yet, but no, quite don't have organs. We have a group of nerve cells that respond to very simple things like touch and gravity and light. We have a group of ringed muscle cells with actin or contractile fibers, those fibers that allow the amoeba to stretch out, right? The actin fibers along the inside of the cell membrane allow it to stretch out without, exploding. And they can take these and they can move these in a rhythmic, so they can move. So jellyfish are kind of one of those ones that are planktonic, nectonic a little bit. They can move a little bit, but they're really mostly going to be drifting with the current. Now, what puts the members of the cnidarians together in the group and then excludes the ctenophorans is the fact that, unlike the ctenophorans with their sticky things, cnidarians have stinging cells, things called nematocysts. Well, excuse me, the cell is called a cnidocyte. Where's my pen? Here we go. Cnidocyte. Inside of that is something called a nematocyst, right? So it's a specialized organelle within the cnidocyte. The cnidocyte is the cell. The nematocyst is the specialized organelle with a trigger. So these cells are going to be on the outside of the organism, and in that particular case, they're going to be used for defense. They're really good for that. As well as for prey capture. So on the tentacles of, like on the hydra, there are these little triggers, and when a prey comes along and it trips that trigger, the nematocyst shoots out this thread. It's kind of like it's coiled up inside of there, and it shoots out and buries that thread into its prey. And not only does it have the thread, but along the thread there are these recurved little spines. So it's not very big, but it's very efficient at capturing small prey. And of course, there's also a toxin that can be introduced as well. Now, if you've ever been stung by a jellyfish, you know what I'm talking about. Hydra just does that more like on the microscopic level. So they can defend themselves this way. That'd be a pretty good effective defense mechanism. Or they can use their tentacles to wrap up prey and then sting them with those cells that can secrete a paralytic toxin, paralyzes the organism, and it holds on to them. These little tiny, can you imagine? It's not one nematocyst that's going to discharge. It's going to be a bunch of them, right? So it's kind of like a spider web. Right? It's not one thread that holds it all together. Very cool. Hi, Olivia. Well, what goes on in this gastrovascular cavity? Well, unfortunately, in the cnidarians, if you happen to have been born one, your mouth is your anus. You only have a gastrovascular cavity. You have a single opening. And that gastrovascular cavity does lots of things. Of course, it brings in food and allows digestion on the inside. More efficient that way, right? Unlike our fungi friends. Diffusion can occur across the gastrodermis, any thin tissue layer, especially if it's highly vascularized. It's got a lot of, blood vessels in it, not in the case of the cnidarians, but any thin tissue, diffusion can work, osmosis can work. Waste, obviously that's diffusion going the other way, dumping stuff into the gastrovascular cavity and then out through the mouth/anus. Gastrovascular cavity, just like in the sponges, gametes can form along there and then out through the mouth anus. Jellies can close their mouth anus and trap water in their gastrovascular cavity and use it like a hydrostatic skeleton. Remember I mentioned that the other day, a hydrostatic skeleton, like if you put your thumb over the end of a garden hose, you know how the garden hose would stiffen up. So they could use that to form and change shape. So they water in their gastrovascular cavity, close up the mouth anus and increase the pressure, and they could change their shape if they needed to. So jellyfish is obviously a good term for them, isn't it? They're very jelly-like. Two basic body forms of jellyfish. There is the polyp form, which is the like in the little picture, the sea anemone. So the picture down below, the artist drawing there, that the one on the left, the polyp form, the mouth faces up, and the Medusa form, named after the, what was she, Greek? I don't remember, with the woman with the snakes on her head. The Medusa form is the typical jellyfish where the mouth faces down, the mouth/anus faces down. The polyp forms are generally sessile. That means they're going to settle down and not move. So that's going to be anemones and corals and things like that. The Medusa form is going to be in the mouth down, and it's usually going to be a floater. Maybe it can move a little bit, but it's mostly planktonic. Some organisms in this group actually have both of these during their life cycle, right? Just like we talked about in the sponges, dispersal is important. So there are some organisms, and if you're thinking, I'll bet it's probably the sessile ones, if any of them, you'd be on the right track. If you're sessile, one of the ways that you could disperse is form a medusa that floats off as well, right? Get away from your parents. That's what that's saying. So speaking of all that and dispersal and reproduction, first let's go simple. They can asexually bud, like in that picture of the hydra down below. That's a little mini hydra coming out there. So they can asexually reproduce. There it is. A little bit longer right there. So they can asexually reproduce. There are separate sexes. Now, dioecious was the term we used in plants. Another term that we generally, I'm trying to remember if this is the rule or not, but gonorchid also means male or female. And I think gonorchid is a term that we generally leave for animals. So I think dioecious can be used to refer to animals too. So a plant like cycads could be dioecious. Jellyfish here can be dioecious, but gonorchid or gonorchism, just another term that means that there are male and females. And male and female separately, right? So it is like the cycads. Of course, there's male and female pine, but they're on the same tree. All right, let's look at this reproduction of Obelia. You're going to look at Obelia in the lab. Obelia is a small colonial cnidarian, grows like in the picture you see there. And Obelia grows with these stalks. And at the end of these stalks, there are these polyps. Remember, the polyp is the beginning with the mouth facing up. Obelia has two types of polyps. There's the feeding polyp that's got the tentacles, and that's easy enough to understand how that works. But there is another type of polyp, and this speaks to that dispersal thing, that produces a bud, a medusa bud. Looks like a little tiny mini jellyfish, and it is. So this medusa disperses, floats with the currents, floats and floats and floats. Eventually, the gonads in the medusa, there's male and female of them, produce the egg and sperm, which are going to disperse. So we're increasing dispersal. We're increasing the mixing and fertilization and so on and so forth. In addition to that, once the zygote develops, you become a larval form known as a planula. There's a planula in the lower right, and you float until you eventually find a place to settle down. So if it hasn't become obvious to you right now how important it is for organisms to disperse, then I don't know what else to say, because they go through an awful lot of effort to do that. Those in the upper right, that little picture, I'm trying to look at that and remember what, those are some of the little medusa buds of some of the Obelia that are split, I believe, are those. I have to look those up again. I can't remember. Coral species that we're going to talk about have the polyp form, and they form that exoskeleton of calcium carbonate. We'll talk about coral here in just a second. We'll talk about different groups. Okay, in red, the major evolutionary innovation that we see in these organisms is the fact that we have the extracellular digestion, like the cnidarians, but the fungi, but it occurs on the inside with the cnidarians. The digestion occurs in the gastrovascular cavity, right? So it's much more efficient because inside that cavity the food is concentrated. So it's just an efficient way to absorb food particles through phagocytosis if you got them all trapped inside of this little gastrovascular cavity. Doesn't mean that the fungi got it wrong. Remember, they're doing quite well. As a matter of fact, all this rain, there's a bunch. If you need a fungus for your field notebook, this is a good time to get it. Okay, let's look at some groups of cnidarians, or what we call clades. Remember that word clade means, it kind of depends on where you're talking about. So let's just call them clades or groups. There's the medusazoa. That's the biggest one. Those are all those that produce a medusa. So this is, any of these other groups all collectively that make medusa forms, at least all their lives or sometimes. There are the Cubozoa, which are the box jellies, very strong swimmers. Chironex fleckeri in the upper right, that's the box jelly. Their stings on some species are fatal to human beings, more venomous than a cobra. They live in Australia, the ones that are that way. There are box jellies in other parts of the world, but the ones in Australia are known for having being some of the most venomous in the world. The hydrozoas, which are the hydra, the hydroids. If you've ever gone snorkeling and maybe in shallow water you swam through some of the turtle grass that was floating on the bottom, you might have felt yourself itching later on. That's because a bunch of little hydroids were stinging you. A bunch of little practically, well, yeah, microscopic little hydrozoans were stinging you with nematocysts as you floated by. There's the Portuguese man of war, beautiful organism, very bizarre critter. It's what we actually call a siphonophore. It's what is known as a colonial organism. Without getting into too much detail, the cells that make up the sail, that part at the top, these things get pushed around by the wind too. So they take advantage of the currents and the wind. And the cells that make up the tentacles, and then I believe it's the cells that make up the reproductive parts, all kind of work together loosely. So it really sort of, again, kind of stretches our idea what an animal is when it comes to tissues and things. So this is kind of like a Volvox. This is like the animal's version, outside of the sponges that we talked about, of again, kind of the Volvox strategy, where instead of cells giving up their autonomy, and I really don't know how to explain that, giving up their sense of self, they collectively get together and form this critter. It's very bizarre. It's what we mean by a colonial organism. Hydroids exist in both the polyp and the medusa forms. The hydra, of course, is only the polyp. That's the only, they don't have the medusa. Obelia has the medusa. Hydra does not. Hydrozoa also have a freshwater species. The Scyphozoa, this is the only class as hydrozoa, by the way. The Scyphozoa are the typical jellies. Most live as a medusa, the moon jellyfish you see in the picture in the blue. The ones down below are really sort of an interesting type of jellyfish. They're kind of a mix between a medusa and a polyp. The one in the little picture, the brown one there, this is one we have here in Florida. This is the upside down jellyfish. That's actually the tentacles facing up. So it's a medusa that's actually evolved to live the other way. You can easily get stung with one of those because the little tentacles hanging up like that. See them all over the Keys. Cassiopeia. Coral sea fans, sea anemones, the other type of cnidarians are called anthozoans, right? And they only have the polyp form, although this polyp can live solitary like this sea anemone does by itself, or colonial. That's what corals are. A coral reef is a bunch of little coral animals living together. Each one of those has its little tentacles for gathering prey, and each one of them makes a calcium carbonate home around itself. That's what builds up the structure of the coral reef. Okay, now, I mentioned corals before. Let's talk a little bit about how corals make a living. Very important. We only have one intact coral reef in the continental United States. In the continental, not counting Hawaii, of course. In the continental United States, we only have one, and that's off of the Florida Keys. So it's obviously very important that we keep that as healthy as we possibly can. But there's been a problem, as you can see in the graph down there, since the 1970s. The percent of coral cover that's diminished. The coral reefs are dying off, and one of the ways they're dying off is through something called bleaching. You can see a picture of what I mean by bleaching there. The coral on the left, which is ironically called a brain coral, looks like a brain, right? Has a color to it, kind of a greenish brown if you want. But the one on the right clearly has been bleached. Now, what is the bleaching all about? Well, they're not being bleached by bleach, but if we look at coral up a little bit more closely, the picture in the middle, the coral is the transparent jelly-looking thing with tentacles. The little brown dots, those are dinoflagellates. Yeah, you remember those from protozoans, right? They're symbiotic dinoflagellates known as zooantheli. Symbiotic dinoflagellates in a mutualistic relationship, by the way. The dinoflagellates photosynthesize. All right, so while they're living inside of the corals and they're photosynthesizing, just like plants, when they make food, they make more than they need. That's why we have something to eat with the plants. So the coral it being an animal, captures food its regular old-fashioned way, but it also has this supplement from photosynthesis. As long as their sun is going to be photosynthesis, that sounds like a win to me. The zooanthellae on the other end of the bargain, they receive carbon, which is very important for living organisms. It's a structural element back from the coral, so they live together. Now, what happens when corals feel stressed? And I know you think, well, how does a coral get stressed? Well, environment changing, temperature out over the reef. Corals tend to be relatively sensitive to things like that. Changes in salinity, salt content, pollutants in the water, disturbance in the water, like what we call turbidity, cloudiness of the water, that really affects their ability to photosynthesize, right? Or at least the zooanthellae. But in this odd thing that we don't quite fully understand yet, when corals feel stressed, they expel, or the zooanthellae leave the corals. They go out into the water. And so the color of coral comes mostly from the symbiotic dinoflagellates. So when they lose those dinoflagellates, they become bleached. Now, is that a problem for the coral? Yeah, you betcha it is. It is definitely a problem for the coral. If a coral doesn't re-assimilate or doesn't take those zooanthellae back up again in a period of time, and it varies and stuff like that, being a species and condition, that coral will die. And that reef will then die, right? Because the coral dies and the rest of the corals die and the reef eventually dies. So there is the ability for the corals to recover, but they need to do it in a certain amount of time, because the corals alone, because they've developed this symbiotic relationship throughout the eons, right? They have become to be dependent on the zooanthellae. So even though they trap food, they don't make enough of a living without the zooanthellae. So they got to have them. It's kind of like lichens. Not quite like lichens, but it's kind of like that. That's what's happening down the Keys, by the way, because of some of the pollution, especially a lot of the fertilizer pollution that runs off from the agriculture down in the southern part of Florida. If you haven't had a chance to go do the snorkeling or diving the coral reef down off the Keys, please do that, especially before you end up going off to school, maybe getting a job at someplace else, because it's absolutely beautiful. All right, where are we? Oh, the bilatera. So now we're going to talk about critters that have left and right and up and down. Most of these critters, not all of them, most of them have a complete digestive system. So they're going to have a mouth and they're going to have an anus. We'll come back to the deuterostomia at the end, but down here with the protostomes, these are all protostomes, these two groups, the ectozoa, which we'll talk about later, and the Lophotrochozoa. So this is the group that's unified together by having one of these two types of larval forms. The Lophophore feeding structure, or not larval forms, but they either have the Lophophore feeding structure or the Trochophore larvae. That's what unites them together. So let's go into them first. I think the first one we're going to look at is the flatworms right here. Flatworms. Now, here's a little trick in understanding the roots of words. Platy, P-L-A-T-Y, means flat. Platypus. Okay. Helminth, if you went to the vet and he gave your a dog an anti-helminthic, or if your horse needed an anti-helminthic, it would be a dewormer. So platyhelminthes literally means flatworm. And so whenever you see that word platy again, think about that. And helminth, also worm, platyhelminthes, the flatworms. Weird group. Soft-bodied critters, ciliated, found everywhere. This critter here, for example, this planarian, is very often found in soil, so terrestrial. No body cavity, so if you cut this guy open, all you would have the digestive cavity, but you wouldn't have that body cavity between the ectoderm, or the outside, and the digestive system. Now this guy, platyhelminthes are an exception. They have incomplete digestive cavities. They have a pharynx that we'll look at it on the microscope, but here's the pharynx in the picture, which is basically a tube by which they bring in food, and then it's muscular, and they're able to mash it up. They're able to mash up their food. Incomplete digestive system means mouth and the anus are the same thing again. There's also another type of incomplete digestive system we find in the parasitic species. Something that's kind of interesting about parasites, if you think about it, a parasite, especially like an endoparasite living in the digestive system of a host organism, really doesn't need a digestive system, does it? As long as it has a way to suck up those nutrients, it doesn't really need to do the digesting itself, which, you know, Mother Nature might favor. That's an easy way to make a living. Okay, we'll talk about those later. Parasites are something to me I find kind of fascinating, but let's go with the why be flat. Why are these organisms flat? Well, if you think about the way these organisms have to interact with their environment, what I mean by that is moving nutrients across, moving oxygen across, carbon dioxide, moving waste products, all those things that you think about if you're thinking, gosh, we talked about this in mosses. We did. And remember, mosses are able to get away with not having vascularization because they stayed small. So there's not a lot of thickness to them. In the case of platyhelminthes, and I'll let you work out the math yourself if you want, but if we take the same volume and we stack it as a cube versus, what's that, like an ice cube tray, the surface area to volume ratio goes up. See, 2-to-1 to somewhere around 3-to-1. And remember, back to Bio 1 again, cells are small because they want to maximize their surface area to volume ratio. That makes them more efficient moving things across their membrane through osmosis and through diffusion, right? So now we're seeing this at an organismal level. Platyhelminthes have evolved to be flat, kind of like that worm you see, right? Well, not worm. That's actually a mollusk right here. This is a nudibranch, kind of like that critter as well. Kind of like all the membranes that are in the thylakoids of a photosynthetic cell, and in the lower right, kind of like the way in your digestive system, the cells that line your digestive system have these little fingers that stick out everywhere, and kind of like the way the mycelial spread out into the environment, they're maximizing their surface area to volume ratio. That's what the stack of membranes does in the thylakoid. That's what those little villi and fingers that stick up in your intestines do. They maximize their surface area to volume ratio. Very simple. Let's cover an area with cell membrane that winds back and forth, so you can calculate the cell membrane, room to move things across, versus if the cell membrane didn't wind back and forth, right? Simple math is going to tell you the one with the winding back and forth has got more membrane to it. So that's why be flat. You be flat, you jack up that surface area to volume ratio, which means you can rely on diffusion and osmosis, which means you don't have to evolve a circulatory system of any consequence. You don't have to evolve an advanced circulatory system. So why do that? Two groups, one much smaller group, this word is a real tongue twister, Catenaluda. What a word. Anyway, only about 100 species, thank goodness. These are the chain worms. There's one on the right. Reproduce asexually simply by budding. And bloop, these offspring at the end of a chain worm, they bud before they detach. Now, that whole idea of budding off as far as reproduction is also found in the next group, at least in some species in the next group. The next group, it contains the ones, or at least one of the ones that you might be a little more familiar with. This is the Rhabdidophora. It's a little bit easier word to say. The Rhabdidophora is a diverse group of about 20,000 species, including free-living ones like this one down below here. This is a planarian. It's this thing with the scalloped head. This is actually a Florida planarian, right? There's U.S. This is one that I've seen. I saw one on my back porch a while back. So look at the head. That's what I mean. Not a worm, not an earthworm thing. It's a flatworm. And then the other one is the tapeworm. These are tapeworms. And tapeworms, notice, have these divisions. It's not segmentation the way we've talked about segmentation, but it's kind of like the chain worm. And we'll get back to the tapeworms in just a minute. They lead a very interesting life. Let's go with the free-living ones first. The planarians. Dugezia is the one we'll look at under the microscope, is the more common planarian. No circulatory system, once again, right? It's all about diffusion and osmosis. They do have a digestive system. There's that muscular pharynx. So they bring in food and they masticate it. They start to mash it up with that muscular pharynx. There is some digestion that occurs outside of the body, so a little bit like the fungi. But the rest of it, which is going to increase the efficiency like it did in the jellies, is going to be inside that gastrovascular cavity. Notice it's even called the same thing, a gastrovascular cavity. Parasitic species don't have a digestive system, right? Especially those endoparasitic species that live in the digestive system of a host, you don't need a digestive system. It's already been digested. All you need is a system by which to absorb that food. So, and like I said, we'll get to that when we start looking at some of those parasitic ones. But if you think about it from an evolutionary point of view, that makes sense if you're a parasite. Evolution, be more beneficial if you don't have to devote any resources at all to a digestive system. Remember I said the other day, organisms bring in energy, and it's like a budget, like a money budget. You spend that energy. So if you're a wind pollinator, is the example I used in class, you're not going to make a big flower. You don't care about attracting pollinators. You just let the wind do it, and the wind doesn't care how big the flower is. Simple nervous system. Oh, an anterior ganglion, cerebral ganglion. Anterior because we talked about advantages of being bilateral. A ganglion is a collection of nerve tissue. So it's a brain-like object, right? Not really a brain, but your brain is nothing more than a collection of nervous system as well. Eye spots by which they can sense light and dark only. Some are hermaphroditic. They can reproduce sexually or by fission. So a little picture on the right-hand side where in the laboratory, they've induced these planarians by making these incisions to split, and they'll split into two complete separate organisms, kind of like the sponges a little bit, right? Their excretory and osmoregulatory system. Well, first of all, let's figure out what an excretory and osmoregulatory system is. Excretory is getting rid of waste products. Your kidneys would be part of your excretory system. Osmoregulatory system is making sure you don't absorb or lose water, right? So depending on what kind of environment you're in, if water is moving into the body, you want to have like those contractile vacuoles, right? You want to have some way to get rid of that. So osmoregulation in general, and our kidneys are osmoregulators as well, are going to keep the body at a rate where you're not losing water or gaining water, right? All that stuff from Bio 1, again, is important. How do planarians do it? Well, they do it in this beautifully simple way with something called a protonephridia. Now, first of all, that's a cool word. Nephridia, if you went to a nephrologist, you'd have a kidney problem. If you have kidney issues, you go to a nephrologist. The study of kidneys and their functions and their structure is nephrology. A proto means a beginning, right? An early kidney. So the protonephridia is an early kidney. Protonephridia is a network of these little tubes with these things called flame cells. And I'll get to the flame cells in just a minute, but put that aside. The bigger chunks out through the mouth. All right, the bigger chunks are excreted into the gut and then eliminated out to the mouth. But back to these flame cells. Look at this thing. It's so cool. Here we go. So there's the planarian. The yellow part is the tubules of the protonephridia, and they're extending throughout the body of the organism. On the end of these little tubules, kind of work backwards in the picture, there's an opening to the body wall. That's where the waste product is going to be expelled out. But the way the waste product is collected is through these little flame cells or flame bulbs. So there's a cilia inside, and that cilia is beating back and forth. Now, what's cool about this is the name flame cell comes from when they viewed them underneath the microscope, they looked like the wick of a candle, right? Flickering back and forth. So these cilia are flicking back and forth. That's causing water to move. Same thing, stentor, right? Water's moving out from that cavity into those tubules, then out through the body wall. So that's how they get rid of waste products, and that's how they maintain water balance. So they're either going to increase or decrease that water flow depending on water balance, osmoregulation, and the need to get rid of waste products. Is that cool? That's so neat. It's a very simple system, right? It takes care of very basic physics. That cilia flicking back and forth creates a vacuum every time it flicks back and forth, and water rushes into that vacuum. That's how a pump works. Absolutely brilliant. Ooh, the parasite ones. Parasitic ones. About half of the rhaptidophorans, so the small group was the chainworms, which I'm not going to try to say the word again. About half of the rhaptidophorans are parasitic. There are the flukes, the trematodes, which have all kinds of wonderful things by which they hold onto their host with, suckers, anchors, and lovely little hooks. Sometimes with multiple hosts, like the blood fluke there on the right. About 200 million people a year are infected. Blood flukes are common among folks who live in areas of the world, very often in Asia, that tend rice paddies. If you know anything about the way rice is grown in a flooded field, so cuts or nicks or openings in the skin of these rice paddy, or the workers out there in the rice paddies, the larval form you see there can actually infect through the opening. They then move in through the bloodstream and actually reproduce. There's the male and the female, the mature flukes inside of the human host, and then producing these ciliated larvae. Now, how do the larvae get out of the host? Well, host defecates. And in areas of the world where, they grew rice in these rice paddies, at least historically, many times the fertilizer that was used was human excrement. So that's how the cycle was repeated. The ciliated larvae would then infect the host like a snail. And this is the interesting thing about some of these parasites. Very complicated life cycles. I was studying one in South Carolina that had a bird. This is in its cycle, this one fluke, a bird, a small fish, and a snail. So the larvae out of the fish the larvae out of the snail would infect the fish. The fish would be eaten by the bird. The parasite would move from the eaten fish into the bird. The bird would then defecate into the water. The larvae would, or another larval form would form and infect the snail. Fascinating, especially to think how that all evolved together. Tapeworms. Lovely critters. Actually kind of fascinating in a way. No digestive system. They're simply going to live in the digestive system of the host, where all of the digested soup is, and use diffusion, for they don't need a digestive system. They're going to use diffusion to take up food. If you dip them into a soup of nutrients, that stuff's just going to flow in by simple diffusion. So why do I need a digestive system? It's ABC, man. It's already been chewed. All I need is something to hold on with, right? Like this they don't have a head, but in the scolex region, see in the picture down below, they have suckers and they have hooks. Those little hooks are the things that embed themselves into the intestinal wall of the host. So this is why if you're thinking about using a tapeworm for weight loss, by the way, that won't work. But the bad effect is as those tapeworms reproduce and move around your intestines, those hooks and those suckers cause damage to the bleeding to the intestinal wall. Now, again, no digestive system, but man, we're still going to reproduce. And the way they reproduce is kind of like the chain worms. They use these things called proglottids, right? And if you see in the picture down below, you got a scolex, a head, with a bunch of proglottids. And each proglottid is the same. See the picture on the right, the one in the purple? This is a microscope of a proglottid. It is a complete hermaphroditic unit. It's got male and female parts to produce a sperm and egg. It can fertilize other proglottids. It can fertilize itself. It can do both of those. But what it can do is make lots of sperm and egg and have lots of these proglottids. And the proglottids are formed continually and then shed in the digestive system of the host. So the scolex stays there, and these proglottids just bloop, and then when the host, the feces of the host, bloop, these things drop out, and these proglottids then reproduce and form eggs. Other organisms, especially browsers, come along and feeding along the grass and the dirt. There we go. That's how you get a tapeworm. And it can occur in beef or occur in pork. This can be a frequent human parasite, especially if you're eating uninspected or rare meats, obviously. Not a good weight loss method. I know that seems weird to say, but back in the turn of the century, well, 1800s to 1900s, it was in some circles touted to swallow these little weight loss pills, which were basically tapeworm eggs. They're not going to absorb enough nutrients to really help you lose weight, but they will do things like clog up your intestinal system and, like I said, tear up the wall of it too. So I don't know if vanity's worth that much. Ooh, this group, the rotifers. I only got one slide on the rotifers, and I debate sometimes about deleting them, but I really shouldn't for a couple of really cool reasons. First of all, let's get the more mundane stuff out of the way. They're pseudocoelomates, so they have a body cavity, but remember it's that technicality, but not being lined by mesoderm. And even though they're microscopic, there's a picture on the right of one, and you're oftentimes confusing thinking they're a protozoan. They're not a protozoan. They're actually an animal. They have highly developed internal organs, a complete digestive system, so what we call an alimentary canal. We sometimes call them wheel animals because of this thing called the corona. And if you look in the picture, you can see where the wheel comes from. The corona, they're filter feeders. So they use cilia, once again, to move the water around, and under a microscope, it kind of looks like it's spinning. So that's where the wheel animals comes from. The reason why I can't really throw these guys out is because rotifers are interesting because they're one of the organisms that can engage in this really bizarre type of reproduction known as parthenogenesis, which is asexual reproduction. Essentially, eggs develop without fertilization. Now, you think, okay, well, this is something that's exclusive to like protozoans and maybe some prokaryotes do think, well, not prokaryotes don't make eggs, but some simpler animals, if you will. But parthenogenesis occurring in rotifers, what we basically are saying here is that there are some species of rotifers, like the Deloid rotifers, the B there is silent, about 360 species that are all parthenogenic. And they've been that way for well over 100 million years. Females are producing offspring without males. So the big question, and I highlight with an arrow there, is where does the genetic diversity come from to create this many species, 360, in a relatively short period of time? So Charles Darwin would be, I guess, relieved that even though we answered some of his questions that bugged him, like the Cambrian explosion, we still have quite a bit more. Interestingly enough about parthenogenesis, let me tell you a quick story. It was a sand tiger shark in the Shedd Aquarium in Chicago. Yes, female. A male sand tiger shark had not been in the exhibit for, I believe, a decade. No male sand tiger sharks. The shark had not left. One day, the trainers or the keepers came in, and there were offspring. There were pups. There were sand tiger pups. Not understanding at all how that could have happened. How did this female become pregnant? How did she deliver offspring? Well, there is some cases where fish can actually store sperm. Females can store sperm for later reproductive events, but this is way too long. That's not the case. But come to find out, there are some species of fish, sharks, for example, like and one down below, that's the bonnet head shark. It looks a lot like a hammerhead. It's related to it. And some species of reptiles, like these lizards that you see, in which the eggs can develop without fertilization. This in some species of lizards, there are only females known. There are no males at all. Now, this does not occur naturally in males. They've got it to do it in the lab, but that is not something that we can document naturally in mammals. I said males. I meant to say mammals. Isn't that neat? So males are unimportant in some species. Now, how they survive, dealing with the genetic diversity issue, that's another story. Where are we? The mollusks. Yes. Oh, wow. Yeah, we got to talk about the mollusks. This is a neat group. No other group other than the arthropods has more species. About 100,000 species of mollusks. These are the snails and slugs and octopus and chambered nautilus. A slug and a snail, essentially the slug is a snail without a shell. Many times slugs have lost their shell. There's a really cool picture in there. That's the banana slug, for reasons that are pretty obvious. There are the big size ones like the giant clams that you see in the picture there of 270 kilos. So that's like 500 pounds. The really pretty ones, the blue-ringed octopus up there in Australia, but one of the most highly venomous animals in the world, by the way. Chambered nautilus. Some are really small. Most mollusks, most members of these groups have remained in the ocean. Oh, the squid, too. I forgot the squid. One of my favorite groups of organisms out there, the squid. But they, most of them remain in the ocean, and they also become very important to us as food. Obviously, oysters and squid and all those kind of things that we eat. My mother loves snails. I couldn't stand them. But obviously for pearls, we'll talk a little bit about how the pearls are formed. But mother of pearl, the inside of the shell here, this is the mother of pearl. That's good. They use that in costume jewelry and stuff like that. But there also is a bad side. For example, among a mollusk known as the zebra mussel. That's what it looks like. Pretty obvious why it's that way. The zebra mussel is an Asian species of mussel, and the problem in the United States is it has invaded the United States, doesn't have any natural predators. So it reproduces like crazy, and it grows so thickly on ships and underwater things in the Great Lakes region that it fouls them. It plugs them up like if that were a pump there on the right-hand side, all those zebra mussels would plug it up. It's an interesting little bit of detective work. The way zebra mussels got to the United States, they're native to Asia, but when ships would leave out of the Great Lakes region loaded down with iron ore to take to Asia to be made into steel, They would load down with iron ore, and that's very heavy, sail across the Pacific, drop off the iron ore. Now, there's a lot of weight there. If they didn't have enough ballast weight coming back, and if you've ever been on a ship in heavy seas, you know why ballast is important. You want the ship to be as low in the water as possible. You want a ship to be heavy in the water. You don't want to be riding on top like a cork, especially in heavy seas. So as they brought a lot of real heavy iron ore over, if they didn't have enough to come back, what they would do is flood their ballast tanks. Water. They would take in water from the port they were in and use that as weight to help ballast the ship. Now, when they were taking in this ballast water, guess what else they were taking in? Zebra mussel larvae. So they come back across the Pacific into the St. Lawrence Seaway, down into the Great Lakes region. Oh, more iron ore. Got to dump off this water. All that water comes back out with those zebra mussel larvae. Just so happens they were able to survive. They were first documented in the Great Lakes region. They moved all the way down into the Mississippi River for same kind of reasons, for shipping back and forth, water being moved. While we're on the subject, we have two definitions for species living here from other areas. We have exotic and invasive. They mean two different things. Exotic means that they're here from someplace else. They're not native to here, but they're not a problem. Okay, invasive means they're not native to here, but they're a problem, like zebra mussels. They kill out native species. That's what we mean by that. All mollusks share a similar body plan of three basic parts. I put them in red here: the mantle, the foot, and the internal organs, the visceral mass or the gut. The mantle, if you've ever shucked oysters, that's that layer of skin, if you will, or tissue that lays right underneath the shell. It's a thick epidermal sheet, and it forms a cavity around that visceral organs known as the mantle cavity. If the organism has a shell, if it's a shelled mollusk, then that's where the shell comes from. It comes from that mantle. The foot, which can be modified. The foot can be used for locomotion among the gastropods, the snails. That's what they crawl on, is their foot. The bivalves, the oysters and the clams, use their foot to actually dig down into the sand. They dig a hole and fall down in the sand so they can hide themselves and just leave their little siphon sticking up by which they can move food through. Snails also can use their foot to seal off their aperture, the opening of their shell. Snails, since they live on land, have to be concerned about drying out. So you see in this picture down below, that's the foot of the snail, and he's used it to seal himself off to protect themselves from drying out. And in the cool group, the cephalopods, the squid and the octopus, their feet have been modified into arms or tentacles, if we're talking about encephalopods. Oh, this beautiful little variations on a theme in all these species. Very, very cool. Now, the internal organs, the visceral mass. They do have a body cavity, but the body cavity has been very, very reduced. And what we mean by that is it's shrunk down. The body cavity of a mollusk is not, if you think about an oyster like in that picture, it's not that whole open space. The true body cavity is a little tiny space found around the excretory organs, part of the intestines, and around the heart. I actually have a picture of the body cavity. The little red arrow on the lower left. So this is the gills, the reproductive organs, the digestive system, all through here, and there's what's left of the body cavity. And this is the heart. The heart is like basically in bivalves almost like the worm heart I was talking about. It's just a pump, right? It's like a tube that constricts. The digestive, the excretory organs, the reproductive organs are all in this visceral mass. So if you like to eat raw oysters like I do, that's what you're eating. You're eating that visceral mass. In aquatic mollusks, they have gills, or things that we call ctenidia, that are used for oxygen exchange. But in bivalves, oysters and those guys, the filter feeders, their gills not only do oxygen exchange, but they put a mucus layer on their gills. So while water's moving through, guess what the mucus does? Traps food. Filter feeders. That's why they have those siphons. That's why they want to dig down in the sand and put those little siphons above so they can filter food, filter water through without anything hopefully seeing them. As we talked about the shell, which is secreted by the mantle, made of calcium carbonate again, that very common stuff. Calcium carbonate. The foraminiferans had calcium carbonate. Remember the chalk folks, I was going to say? Folks. What does the shell do? Protection. Kind of hard to chew through something like that. You think about this critter up here at the top, this chitin. He's pulled himself. The chitins can work like those rubber hooks that you put up on the wall, and then you turn the hook down. It creates a suction. Chitins can. Let me go back one slide. Chitins have a foot. There's a chitin right here. Don't confuse that with chitin, the chitin. But here's their foot. They take that foot and they lie up against the rocks and they pull that foot in, creates that vacuum, and they seal around the edges. Very hard for a predator to get them off the rock. So that's dissuades predators. Also for intertidal organisms, right? Tide goes out, you got to seal up, you got to clam up so you don't dry up, right? Some have lost their shell. The slugs, for example, have lost their shells. How does a pearl form? Well, it has all to do with the nacre, or the mother of pearl, that shiny stuff. And if you look in the middle of the slide, and just to the right is actually a picture of a pearl, you can see what goes on. The mother of the pearl, the part in the yellow, is the shiny part. And a little piece of some foreign body, a little piece of dirt gets in there. So what the bivalve does is it continually begins to wrap layers and layers and layers of nacre around that foreign body until you eventually make that pearl. So inside of a pearl is some sort of like a piece of sand or some irritant that made its way into the mantle cavity of this organism. And not all pearls are perfectly round, of course, because they don't form that way. Oh, the radula. We got top. This is unique among the mollusks. Not all of them have it, but only mollusks do have it. Very effective, wonderful adaptation for scraping algae. If you're an algae scraper like snails are, if you ever had snails in an aquarium, you know what I'm talking about. And modifications of a theme again. Predatory gastropods can use the radula and modify the radula, use like a drill, like you see in the picture right below there. That's a hole through another bivalve that some predatory snail, gastropod, had drilled a hole through, and then obviously digested the contents. Conus snails have taken their radula and turned it into a harpoon. That little picture on the right, see the harpoon with the hook, the barb coming back with a venomous gland. There are, I think there's some reported deaths from human beings from being hit by conus snails and their harpoon radula. Now, bivalves are filter feeders. They don't scrape, so they don't have a radula. In cephalopods, the radula has been turned into the beak. Squid and octopus have a beak, and that's what they've modified their radula into. But I want to stick with the radula for a second as a scraping device. Here's a couple of pictures of the radula. In the bottom left is the electron microscope picture. The radula is made of these hard teeth that are scraping teeth. The picture in the upper right might be a little easier to look at. So the snail's head is to the right. The algae is the green stuff. And notice that the radula with these little teeth it's not really moving around like an escalator belt would move, but the action of the muscles, all these cartilage muscles in there, pulling on. Next time you see a snail, if you have an aquarium up against the glass, see how close you can get. See, you can almost you can see the scraping motion of the radula. It's the neatest little adaptation. Oh boy, reproduction in these guys. All right, first of all, they have a trochophore larvae, which is an interesting observation, right? Because so do annelids, which is a group we haven't talked about, but we're going to. Those are the segmented worms, are the annelids. So they have the same kind of larvae as the annelids. And if you've been paying attention all semester long, paying attention to what I've been saying, similarity of development, right, among organisms have given us indication of relationness. So mollusks are relatively closely related to segmented worms. They share a similar type of larvae, the trochophore. All right, mollusk reproduction. Gonorchid, there usually are males and females. There are some hermaphroditic and some oysters that can actually change sex. There are some species of fish that can do that too, by the way, so that's not unusual necessarily. Among many mollusks, especially those as you would expect living in the ocean, external fertilization is going to be the rule. They're going to spawn their sperm and egg. Gastropods who live on land, as you might imagine, are internal fertilizers. right? It's the dryness of the land. It's gonna dictate that. In the larval forms, now cephalopods, squid and octopus have direct development. So a large octopus looks no different than a smaller octopus. It's just the size. That's it. Just size that's different. Among the other mollusks, we have the trochophore larvae, which is found in all groups. That's a free-swimming, larval, drifting, dispersing, get away from your parents kind of stage. And among the bivalves and some marine snails, most snails live on land, there is a second, almost octopus-looking thing called a beluga larvae. That's another free-living larval stage. Both of these, whether you only have the first stage and then you're the adult, or you go the first stage, then the second stage, and then you're the adult, both of these are great for dispersal. Both of these are great for moving around. Okay, 4 big groups of mollusks, the 4 major classes. First of all, the chitons that I mentioned before are the Polyplacophora, mini plates, placophores. Chitons are identifiable because they have 8 plates. 1, 2, 3, supposed to be 3, 4, 5, 6, 7, 8. 8 dorsal plates, 8 calcareous plates. Okay. Now, this looks like segmentation. That characteristic that we said is among animals was very important, but segmentation doesn't exist in this group. That's not true segmentation. That's just the shell is segmented. These are herbivores, grazers. They're the ones that are going to suck themselves down on the rock when the tide goes out and then use a radula to scrape the algae off the rocks. The other group are the gastropods, which is the biggest group. About 3/4 of all mollusks are gastropods. This is mostly a marine group. There are some freshwater species. There are some terrestrial. Well, these are the only terrestrial mollusks. These are the snails and the slugs. We have, of course, many species of these in Florida. This one up above is one of my favorites. This is known as the keyhole limpet, L-I-M-P-E-T, because it's got a little tiny keyhole. These are apple snails, which we have here in Florida. This is a slug, snail without a shell, eyes at the end of its stalk, sensing tentacles, and a pneumostome, which is a really simple reproductive respiratory system, just basically a hole that air moves in through. Now, and nudibranchs in the background, the colorful ones are the nudibranchs, but we'll talk about nudibranchs in a couple slides too. Nudibranch literally means naked gills, so these are, again, slugs, but they're slugs that live in the ocean, and the colors in the background that you see of the nudibranchs aren't fake. They are very cool, colorful critters. But first of all, let's talk about torsion. Okay, what do we mean by torsion? Snails, all right, the one in the lower left, that's the wolf snail. That's another Florida species there. Snails, the way their digestive system is set up, if you look in the picture in the upper right and go all the way to part C over there, the anus of a snail dumps its contents right out over the head of the snail. And the little furry thing that's sticking out there in that picture in part C, that's the gills too. So the anus dumps this defecate material out over the gills and out over the head. Now, this causes something called fouling, as you can imagine what we mean by that, right? So from an evolutionary biologist point of view, the idea is, why would organisms do this? You know, if you look at the torsion and gastropods, why would something happen? And this happens during embryonic development, whereby the entire digestive system, the thing in the green, twists itself around. That's the torsion. Twists itself around so that the anus dumps out over the head and the gills and causes fouling. What's the point of all that? Well, we think as gastropods evolved from a very simple sort of flat shell that you see there to one that was longer but asymmetrical in weight, that it became easier to move the shell if the shell twisted, right? See how in that picture? So that would cause the digestive system to twist as well. In addition, what they found is the torsion in the gastropods allows the gastropod itself to much more efficiently not only or withdraw or completely withdraw into the shell of the organism to protect itself, but they're also more efficient at gill ventilation when the gills are in front. Now, is fouling still an issue? Yes. But the reason why I think this is an interesting topic to bring up this demonstrates that, remember what I said in evolution, that evolutionary trends, evolution is a compromise. You don't get everything you want. So does this cause fouling? Yes. Are the benefits of twisting the shell when it comes to ease of carrying it, right? You can see it's a lot easier to carry that asymmetric shell along the long axis of the snail, or withdrawing your body, or for gill ventilation if you do cause fouling. So it's a trade-off, right? In a perfect world, you wouldn't have this fouling, but it's a trade-off, and organisms always go through that. As I said, evolution is compromises. You can't change what you are. You have to kind of work with what you had. Or as my old man used to always say, you got to dance with the one that brought you. So this is the way it is, so this is the compromise. Tons of examples of that out there in the animal world. The nudibranchs. Marine slugs, for the most part, brilliantly colored organisms. Many of them are active predators. They get their name from the fact that see the yellow and this purple one. Isn't that cool? I've seen a couple of these when I was scuba diving. They're so neat. And these are all gills, naked gills. Nudibranch. Branch is gill. Fossil records suggest that they did have a shell, but lost it during evolution. Well, here's another one puzzling. Seems odd. Why would you do that? Now you're this brilliantly colored, soft-bodied critter without a shell. That one down the lower left, a lot of these appendages can be modified. Number one, the little appendages that are sticking out can increase the surface area for diffusion, and that critter down in the bottom. Some of them can have chemosensory cells that act like rhinophores, so they can smell, essentially. So neat. But back to the color thing again. Why be so brightly colored and yet not have a shell? Well, if you have a defense mechanism, you may want to advertise that. Okay, years ago when I first moved to Florida, I worked for a burglar alarm company. And I would go put burglar alarms in people's houses, and they'd say, well, I don't want to put a sticker up because I don't want to tell the burglar that I have a burglar alarm. I'm like, well, yes, you do, because you want to put the sticker up at the window. Keep him out before he gets in your house. Well, same kind of thing is happening with these nudibranchs. They're very colorful because organisms that would generally want to feed upon them have learned that these brightly colored critters, for one thing, either don't taste very good, so they're very distasteful, or some of these guys, since they're predators, they feed on cnidarians, and they take the stinging cells, the nematocysts, right? And they don't digest them. They migrate throughout the body until they end up being collected in these projections, these serrata, these yellow things, these things that are sticking off the nudibranch, or just in the skin in general. So a predator trying to eat a nudibranch going to get a mouthful of stinging cells, right? This is what we call apismatic coloration, warning coloration. It's the reason why wasps and bees have the black and the yellow. It reminds critters, don't mess with me. Why the South American poison dart frogs are so brightly colored, because they secrete a poisonous substance on their skin. Coloration is so important in critters. Like, for example, that butterfly. That's not a warning coloration, but among species of butterflies, there are two species, the monarch and the viceroy. They look very close to one another. Now, the monarch, is unpalatable to birds, but the viceroy is not. Viceroy birds can eat. Birds have learned to avoid the monarch. Don't eat that one that looks like that. Viceroy butterflies evolved through time to look much like a monarch. So the birds go, oh, I don't know, maybe that's a viceroy, maybe that's a monarch. My guess is the birds aren't that good at identifying butterflies. They stay away from them. Sometimes it's good to advertise that you're a badass, I guess, right? The filter feeders, the bivalves, those that have two shells. That's what we mean by bivalves. Clams, scallops, mussels, oysters. Most of these are marine. There are some freshwater species. The way the shell works in these bivalve organisms is the shell is two hinged valves, right? Two hinged sides to it. And up here at the top, there is a ligament, up here at the top, right here. There's a ligament that if the shell were left to its own devices, that ligament would cause the shell to open on its own, right? Now, what the organism does is it uses these two adductor muscles. There they are right there. In A&P, anatomy and physiology, or in terminology, adduction means to do that. So your legs, when you bring them together, you're adducting, and then when you spread them apart, you're abducting. So adductor muscles pull things together. So that hinge is causing the shell to open naturally, that hinge ligament. The adductor muscles are keeping the shell closed. And when the tide goes out, those adductor muscles keep that shell closed very tightly. And when organisms are trying to attack that organism, or trying to get in there and eat this visceral mass, see there's a mantle in the foot. The labial palps, those are cool. Those are the things that actually help guide the food into the mouth, the food that's transported over the gills, right? So water moves into the inhalant siphon, across the gills, oxygenation, food, into the mouth, out through the excurrent siphon. Where, no doubt, or where you wouldn't be surprised the gonad and the anus dump into as well, right? So the shell is stayed closed. When the shell goes out, when the tide goes out, and when the tide comes in, opens back up again. These are filter feeders, so they don't have a radula. There's really no head. And as I said, they have those labial palps that they use to filter food into their mouth. The picture of the sea otter is an interesting. Sea otters have learned to crack open the shells of oysters and eat the inside of them. That's why they always say, who would ever eat an oyster? Who was the first one to eat one? Historians or anthropologists tell us that what that was many times human beings would watch animals, and animals ate it. They say, well, it must be okay for us to eat it. So I imagine maybe Northwest, United States folks watching otters eat oysters suggests that you could eat this thing that looked really bizarre. Oysters, but the sea otter takes a rock, smashes against the bivalve or the oyster till he cracks the shell open. So he doesn't worry about those adductor muscles and holding the shell closed. He just beats the shell open. Cute little critters, right? The cephalopods. Ah, yes, the head foot critters. All of these are marine. They have a closed circulatory system. So unlike the other especially the bivalves with their open circulatory system we talked about the other day, these are the only ones. So they can get a little bit bigger, higher metabolic rates. Their foot has been modified into arms or tentacles. Now, there is a technical difference between them. Arms have suckers along the length. So octopus have nothing but arms. Squid have tentacles, and those have suckers at the end. So an octopus has 8 arms. Squid has 10. 8 arms, 2 tentacles. Amongst the squid, too, oftentimes these tentacles, especially the suction hooks, can also, or the suction cups can actually have hooks in them. They use to hold on to their prey and defend themselves. So that's pretty nasty critters. Beak-like jaws, toxic saliva. This is what their radula has been modified into, especially in the purple ringed octopus that I talked about. That's where the toxin comes from, is the saliva. And of course, these are the critters that have gotten the bad PR, the bad press from, ancient seafaring stories about giant cephalopods attacking ships. And so for many years, we thought it was nothing more than just sea stories. It wasn't anything like that, but come to find out, as we go through this in just a second, you'll see there are some of these large critters out there. All right, how do these cephalopods get around? Well, they have a mantle cavity that they suck water into, right? And then they propel, they shoot that water out through a siphon, and they use it for jet propulsion. And they can also use it to spread the ink, which, you know about cephalopods spreading the ink. It protects them. There's a diver in that picture right there on the left. All you got to do if a predator's coming after you is make him just stop for a second and look someplace else. No internal shells in these guys except for the chambered nautilus, that critter that you see in the lower left. Very neat organisms. In the other cephalopods, the shell has become internal. It's become something called a pen, P-E-N. But we do think that ancient cephalopods, or things we call ammonites, did have shells. And they were huge critters. There's a picture in the middle of what the ammonites have. We all went extinct with the dinosaurs. One really curious thing about these organisms is their ability to change colors using chromatophores. They use this for camouflage, and they use this for communication. They could use bioluminescence for breaking up their silhouette. So down in the deep water, it's hard for predators to get an angle on them. Highly developed nervous systems. Those are cuttlefish in the lower right. Very intelligent, interesting critters. Now back to their size again. Where are their big squid? Well, yeah, we knew there were because they would wash up on the beach every once in a while. And up here on the right, these are sucker scars on the skin of sperm whales. Sperm whales love to dive deep and feed upon these squid. Well, before we even saw the squid alive, which was just recently, that picture in the middle was a picture of a giant squid just taken not too long ago, we would see these sucker scars from the hooks on the suckers that I was talking about, and they were 5 centimeters in diameter, which gave us an indication these animals were somewhere 20, 30, 40 feet long. Now, we can estimate they get as long as 60 feet. So it makes you wonder what other types of deep sea critters are out there in the ocean that are there that maybe we just haven't seen. Smart critters too. Intelligent animals. Put the prey into an aquarium in a jar and put the lid on there, and then try to get the see if the octopus can open the lid. It can not only open the lid, it can unscrew the cap as well. A lot of reports of divers on certain areas where there is an octopus, and the octopus develops a relationship with the diver, kind of like a pet. They come out and they like to be petted and everything. They're fascinating creatures. I think if I studied invertebrates, it probably would have been them. Sadly, mollusks are one of the largest groups of organisms that are suffering animal extinctions, and again, that has mostly to do with habitat destruction that occurs. I only got a few minutes, but I do want to get into the annelids, so we can pick up with that next week. So let me go ahead and start that. The annelids, so we're down here. These are the segmented worms. Found all over the place, diverse habitats. There are marine forms, there are terrestrial forms. Typically, the body plan is they have a head with, again, that cerebral ganglion. Some have eyes. Earthworms don't have eyes. They live subterranean, don't need eyes. If you don't need something, don't create it. Eyes are expensive, right? And here segmentation is taken to its highest degree, right? These organisms are known by their segmentation. Divided on the outside, divided on the inside. Segmentation allows compartmentalization, allows them to specialize, allows redundancy. I said the other day about kidneys in each of the segments. The little white lines, those are the kidneys. And they're repeated in every segment. Now, just for fun, if you want to know what the world's largest earthworm was, there it is. I think it's in the tropics somewhere. All right, so as I said, specialization of the digestive system. One of the things you can do with segmentation. So on the front, you can have the mouth. You can have then followed by the esophagus, the crop, the gizzard, the intestines. All those things I'm describing in that picture can be used to specialize. So these organisms can become specialized in their function in their environment. The segmentations in the body construction allows for a hydrostatic skeleton. They can use alternating muscle contractions to squeeze out and pull themselves up. Along the edges, they have these chitinous materials called setae. A couple of different spellings for setae, S-E-T-A-E. C-H-A-E-T-A-E, but that's the little bristles. If you ever felt an earthworm, you know that you feel these little bristling on the outside. Those things are made of chitin, and they help the earthworms, stick get some traction in their environment. Closed circulatory system, diffuse across their body surfaces. They stay small, so they can do that. Blood vessel, major blood vessel is dorsal. Nerve cord is ventral. Now, highlighting that in red because that's important later on when we talk about, which group of invertebrates may have gave rise to the vertebrates. An excretory system very similar to the mollusk, so the flame cells again, repeated in each segment. See, now we got a nephrostome, which again is a flame cell with a collecting tubule, takes that material and dumps it out the external opening. All right, so it's 2:29. I'll go ahead and stop right there. We'll talk about the different groups of annelids when we get back together. Remember, which will be tomorrow. And remember, you do have your exam on Monday, so if you have any questions, get those together and be sure to present those to me. All right, that's it for today.